Okay, great. So yeah, final talk before lunch, so lots of pretty pictures, hopefully. So yeah, my name's Chris Cooney. I'm a research fellow and group leader at the University of Sheffield, and just like to briefly tell you about this journey we've been on to study kind of broad-scale patterns of avian colour evolution and the role of computer vision and AI in helping us do that. So yeah, just a bit of background about where the kind of motivation comes from. So personally, I'm very interested in understanding the evolution of sexual traits, and so in birds in particular, they're renowned for their kind of spectacular diversity in plumage coloration. And I find this quote from Alfred Russel Wallace, who kind of specifically identified the kind of pure and intellectual enjoyment that many of us get from studying colour in the natural world. And so I'm very driven to kind of understand, as hopefully you can see from this tanager picture, this kind of very complex and multidimensional phenotypic diversity. And so just another nice illustration of kind of the complexity. I know we all deal with complex data, but in terms of coloration, how complex colour pattern variation can be. So here's a male and female of a North American parulid warbler species. So here's closely related species of birds of paradise that vary enormously in their kind of plumage pattern colour diversity. And if we extend this out to kind of all birds, sorry, probably the details are lost here, we're kind of dealing with an extraordinary diversity and complexity of coloration in birds, but this applies to many other kind of diversely coloured animal and plant groups. And so some of the key kind of research goals that myself and my group are interested in studying or achieving is to essentially find better ways to measure and characterise the true scale of avian colour pattern diversity, and then to use this better quantification to understand the genetic, developmental, ecological and evolutionary causes of this diversity. And along the way we hope to generate highly kind of generalizable and user-friendly tools and workflows for analysing this type of data. And so to get right into where we've come from and where we're at now, so I've been part of a long-term project that's with its aim is to essentially collect a really large-scale data set of avian plumage coloration for the world's birds. And the approach we've taken is to use calibrated high-resolution UV-vis photography of museum specimens. So here's a kind of photograph of our setup at the Natural History Museum in the UK. And here's some kind of snapshots of the type of data that we're collecting. So for every species, we currently have data for 8,500 bird species, multiple specimens per species covering males and females. We take these kind of standardized images in both the visible and UV spectrum, and we take images from 3 different orientations of the specimen in order to kind of capture the full plumage colour phenotype. And so I should say at this point, this work is in long-term collaboration with Dr. Gavin Thomas, who was successful in getting an ERC grant many years ago now, which basically underpins a lot of the data collection I've talked about here. And so the kind of challenge, I guess, is how do we efficiently extract usable and meaningful data from these images? And so in comparison to kind of very nice, clean images of well-preserved tanagers, like the paradise tanager here, we sometimes have very differently preserved specimens that pose certain challenges to extracting meaningful data. So here, parts of the plumage colour are occluded. There's other details in this image, like labels and feet, that we don't necessarily want to kind of measure. And so just briefly, we've explored different approaches to processing this data. One is a citizen science approach under the name Project Plumage. And if you'd like to kind of learn more about that side of things, please visit the website. But the approach I'll talk about today is machine learning. And so there are 2 strands to that. One is simply kind of specimen segmentation. I say simply. And the second one is identifying body regions on the specimens in order to provide kind of a spatial description of colour variation across specimens. And so to jump right in, I should say at this point, the work I'll talk about from now on is effectively not mine. It's a collaborator of ours, a former student, Dr. Yi-Chen He, who's here in the audience. Yi-Chen, do you want to put your hand up? There he is. So all the good stuff's his and any misunderstanding is mine. So first, Yi-Chen, during his PhD, helped us, developed a semantic segmentation model based on the Deep Lab version 3+ architecture in order to try and help us speed up the processing of this data set by segmenting out the specimen from every image. And so just briefly some figures. Yi-Chen generated a training set of just over 1,600 annotated images per view, so over 5,000 images in total. Then we performed various testing of the model performance, and we compared this semantic segmentation approach to classic computer vision methods. And long story short, the semantic segmentation is shown in grey here, and there was reasonably good performance, sort of IAU precision and recall metrics in the 90s. And compared to kind of classic computer vision signal processing methods, it greatly outperformed those, as you might expect. And so just some insight into kind of the successes and the failures of this approach on this data set. So at the top rows in these images are very good segmentations, and then towards the bottom there are less good segmentations. And I've highlighted one in particular on the right-hand side here, where the false positives shown in red, which essentially areas we don't want to reconstruct as being plumage, are in this case the legs of this specimen. And we're finding that this approach will need some refining, because occlusions to the specimen are varied across this big data set, and sometimes we are falsely recognizing areas that we don't want to. And so, as I mentioned, the second strand of this work involving computer vision and machine learning is to automatically identify body regions of the specimens so that we are able to extract essentially point measurements of homologous regions across specimens so that we can more easily compare body region differences in coloration. And so we've identified 10 key body regions that we're interested in locating, along with the reflectance standards that we use to calibrate each image. And so again, Yi-Chen explored a method known as pose estimation using a stacked hourglass design. And this approach has been developed primarily for identifying, or used in human pose estimations for identifying body regions in images and videos on humans, but it can be applied to in other scenarios. And so Yi-Chen developed this model and trained this model to recognize these patches of interest and to generate predictions of the location of these body regions. And so again, here's some insight into the performance. So on the top, we have fairly good predictions of the location of these body regions. And in the lower panel, some less good examples of predictions where, for instance, in the third image down, you can see that the prediction of where the crown is on this kind of unusually positioned specimen actually has located the region on the eye, or what used to be the eye of the specimen, which is obviously not ideal and not correct. But by and large, the performance is acceptable. So if in this radar plot, positions to the outside of the radar plot are good performance, we can effectively identify the standards 100% of the time. And we can also identify certain body regions very, very consistently, but other body regions that may be occluded or more difficult or more subjective actually to identify are located with less accuracy. But overall, we can show that expert labeled images and machine learning predicted images generate very similar color estimates. And so we feel that this is a kind of viable approach. And just in the last few minutes, I just wanted to show how this kind of work has fed into some research studies, some empirical questions that we've been addressing, and one recent paper that we're particularly proud of has basically leveraged the power that Yichen's models have allowed us to address questions on a kind of unprecedented scale. And so one of these analyses involved studying or addressing the long-standing question of whether there are actually latitudinal gradients in the colourfulness of bird species. And so here's the kind of overview of the workflow, and you can see that we've gone from image acquisition through the very important step of image segmentation or specimen segmentation. And once we have that step, we're able to kind of use automated approaches to extract the colour data and map into a kind of relevant colour space for understanding the diversity of bird colour. And so that's known as the kind of bird tetrahedral colour space. And so we were able to do this given the efficiency gains that we got for 4,500 species that we had data for at that time. And that equates to over 140,000 unique images. And so just to kind of visualize what that color diversity looks like, we can pull out samples of avian plumage colors from that data set and map them into a representation of how they'd be viewed by a bird. And so here's a sample of 1 million avian colors pulled out from that data set. And you can see the kind of diversity of hues and colors that exist in avian color space. And so we were able to relate that color diversity or that colorfulness on a species level to the distribution of those species across the world and prove, I would consider conclusively, that there is indeed a latitudinal gradient in colorfulness, and that actually equatorial birds in general are more colorful than temperate region birds. So we're really missing out on a kind of major axis of global diversity by living in the temperate region, most of us. And that if we really want to see the full range of bird color diversity, we should maybe go to Borneo or somewhere like that. And just finally, Yichen has continued to develop these models, and actually a key goal of the group at large is to make these workflows and these tools available and very user-friendly. And so Yichen's been developing a platform called PhenoLearn, which runs on a Python base but has a GUI for user-friendly interaction. And so we're able to do things like generate training sets, actually train models very easily, and then review those predictions and correct them if necessary. And just quickly, this is applicable not just to color-related questions, but also to kind of general morphometric questions. And another part of Yichen's work showed that the pose estimation techniques are very reliable ways of generating very usable kind of morphometric data. And so just to finish, this is obviously not the end of the story, and we're looking to explore different ways of not only segmenting and locating body regions on specimens, but also using computer vision and deep learning techniques to actually extract features of color pattern diversity from the images themselves. And that's something we're kind of working on currently and are looking to kind of finalize over the next few weeks and months. And with that, just say thank you very much for listening.